Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stoliarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our special Virtual Enlightenment Salon of May 22nd, 2022 on epigenetics and gene therapy and the prospects that these fields present for curing diseases, lengthening our lifespans, overcoming some of the greatest problems that have plagued humanity since the start of our species' history. Joining us today, we have an esteemed panel of U.S. Transhumanist Party officers, including our Director of Visual Art, Art Ramon Garcia, our Legislative Director, Jason Geringer, and our Director of Applied Innovation, David Shoemaker. And we have numerous esteemed guests today. We have Klary Niezen-Koning, who is a PhD, a biochemist, a clinical laboratory geneticist, and senior researcher in medical sciences at the University of Groningen in the Netherlands. She is an expert on epigenetics, mitochondrial genetics, and the pathophysiology of metabolic diseases. We also have a returning guest, Liz Parrish, the founder and CEO of BioViva, well known in the longevity community. We are going to hear an update from her on her company's work, and accompanying her is our good friend, Dr. Bill Andrews, President and CEO of Sierra Sciences, one of the foremost researchers on aging, telomeres, telomerase in the world today, and also a good friend of mine and a famous ultra runner. So welcome to Liz, Klary, and Bill. And helping me moderate this panel today is Simon Waslander. All of you who attended our previous virtual Enlightenment Salons on April 17th, and then our Death of Death panel with Dr. Terry Grossman and Dr. Jose Cordero will recall Simon's insights during both of those salons. So I would like to hand the floor over to Simon for some of his introductory remarks and some of his thoughts on the problem of aging and the crucial research that is currently being undertaken to address this problem and hopefully lengthen our lifespans in the process. So Simon, please go ahead.
Well, thank you so much, everyone, for being here. And I'm just so very excited for this, to be on this panel as a co-moderator. And the thing is, aging is the highest factor in 90% of humans in disease. So that makes this very relevant to be listening to this right now. And the biggest problem I personally hear from many advocates and also from people who are interested in the aging space, or just normal clinicians and doctors like, Simon, escape aging velocity, immortality, and curing all disease. That's just so long way off. It's not relevant for me. I'm going to be dead by then. Well, what we're going to discuss today, my friends, is epigenetic markers that can measure aging at a mitochondrial level. We'll be talking about gene therapies and telomerase therapies that have already proven, published, peer-reviewed papers, extended lifespan in mice by 40%. And that's happening today. So this is relevant today. And let's make aging research a priority for us and for humanity. And let's get this show going.
All right. So we have a presentation that Dr. Niezen-Koning has prepared for us. And Dr. Niezen-Koning, you have the floor.
Thank you so much. Well, we just started with this nice slide, mitochondria, oxidants, and aging. And according to the free radical theory of aging, the formation of intercellular reactive oxygen species is the most important driver of lifespan. Well, that's a sentence that is known to everyone, I think. And below in the slide, you see the antioxidant scavenger reactions, and you see the SOD, the superoxide dismutase, that is breaking down the toxic charged oxygen radical molecules. And the other one, aging, increases much of the TRX contents. And you see the ratio of the glutathione and the oxidized glutathione that maintains the intercellular redox homeostasis. Those systems are antioxidant scavengers reactions. Next slide, please. Well, we asked ourselves whether the D-loop mitochondrial DNA methylation could be a nice biomarker for human diseases. And when you look into literature, some diseases are coming up. For instance, cancer. They have done some experiments in which the D-loop of the non-cancerous tissue was methylated and the cancer tissue was unmethylated. In metabolic and cardiovascular diseases as well, the D-loop methylation is linked to an increased risk of diabetes and could be as well an indicator for the early stage pre-diabetes. Aging and senescence, well, then you see demethylation of the D-loop, so no methylated Cs are there found in the D-loop. And in neurodegenerative diseases, especially in Alzheimer's, Parkinson's, or ALS, you can see, well, for Alzheimer's disease and Parkinson's disease, there's not enough evidence, but in ALS there is a decreased D-loop methylation. And we have also other examples: Down syndrome, non-alcoholic steatosis, major depressive disorders, and ADHD. Next slide. But when we look into the biomarkers, I think we need something of the mitochondria, the basics of it. Well, what are mitochondria? Mitochondria are membrane-bound cell organelles. They are the powerhouse of the cells, as they call it. And when in a certain place more energy is needed, the mitochondria can reproduce by growing larger and then divide. They can thus change their shape and move around in the cell where they are needed. The ATP is generated by flow of electrons through the oxygens, and that is called the respiratory chain. Regulation of the calcium homeostasis is one of the tasks for the mitochondria, and the production of reactive oxygen species as well. Next slide. Here you see some imaging data of mitochondria. On the left side you see the electron microscopy, and on the right side you see a nice picture of the outer membrane and the inner membrane and the cristae. The cristae are formed in the matrix, and it gives a bigger space to put some proteins on. You can compare it with the intestine in your stomach, in your body. Next slide. When you look at the respiratory chain complexes and the mitochondrial DNA, we have 5 respiratory chain complexes, and through those complexes, complex 1, 2, 3, 4, and 5, it's not otherwise like that, and of course they have their names, but there is an electron pump going, and that is giving energy, and the energy is can be stored in ATP. The respiratory chain complexes are encoded. Some are encoded by mitochondrial DNA, and there are about 13 complexes that are encoded by the mitochondrial DNA, and well, I think about more than 40 by the nucleus. Sorry, one slide back, please. When you look at the right picture, you see the circular mitochondrial DNA, and you see a part of the D loop as well. The D loop is an area of the mitochondrial genome which consists and is non-coding DNA. Next slide, please. As we all know from literature, the role of methionine and the role of its metabolite is well acknowledged. The beneficial effect of the methionine restriction on lifespan extension and metabolic health is that we have less oxidative stress, induction of autophagy, and less free radical leakage from mitochondria. In the breakdown of methionine, you see its metabolite, and that is S-adenosylmethionine. This is a universal methyl donor, and that's for us at the lab, it's a nice compound to use. The increase of SAM in blood is associated with excess nutrition and accumulation of fat mass, leading to whole body metabolic impairment. Next slide, please. We have used this compound as adenosylmethionine to look whether we could induce the D-loop mitochondrial DNA methylation in human skeletal muscle cells in culture flasks. And we see on increasing concentrations, we see indeed an increased methylation arise throughout the D-loop and the site B. We performed this one with pyro sequencing and has been described in our recent paper this year. Next slide, please. And this paper was about a clinical presentation of patients, because I'm an expert in metabolic diseases at the lab. We had some patients with an unexplained clinical presentation, and they present with all the clinical signs and symptoms of myopathy. And we thought, that's strange, because they didn't harbor any mutations of the mitochondrial DNA or in nuclear DNA during their clinical diagnostic workup for the whole genome. And as a standard routine, we also take muscle biopsies from these patients, and all those patients show decreased ATP generating capacity without any mitochondrial nor nuclear DNA mutation. So that was for us very strange, but also fascinating. Next slide. That brought us to the hypothesis that perhaps another layer of regulation and was the mitochondrial DNA methylation, and that could be a factor leading to an impaired mitochondrial function and the subsequent decline in ATP generation. So next slide. The aim of our study was to investigate whether the observed decrease in that ATP generating capacity in the muscle cells could be explained by change in mitochondrial DNA methylation or other mitochondrial epigenome parameters. Next slide, please. We looked at the site B and mitochondrial DNA methylation via pyro sequencing, and that was higher in muscle biopsies for myopathy patients compared to healthy controls. You can see it. Yeah, I don't have a pointer to explain, but I think you can see it yourself. We have investigated 14 myopathy patients and 5 healthy controls. Please, next slide. And when you look at the ATP generating capacity versus the methylation levels, it negatively correlates with each other in muscle tissue. Next slide, please. But to take muscle biopsies from patients is very invasive, and therefore we thought, well, perhaps it can be measured in skin fibroblasts as well. So we compared the skin fibroblasts, the cultured skin fibroblasts from 4 of our patients with the muscle tissue and the mitochondrial DNA methylation was correlated. There was a correlation, and we concluded the skin fibroblasts could be used as a proxy. Next slide, please. Well, a short wrapping up. We saw an increased mitochondrial DNA methylation in human skeletal muscle cells upon increasing concentration of SEM. The increased mitochondrial DNA methylation in muscle biopsies from patients compared to controls was increased. The average mitochondrial DNA methylation levels negatively correlates with the ATP generating capacity in muscle tissues, and the muscle tissue and corresponding skin fibroblasts showed a correlation in mitochondrial DNA methylation and could be used as a proxy. Next slide, please. Well, when you look through literature, and nowadays it's still the same statement that it's mitochondrial DNA methylation, is this available, yes or no? It's still a matter of debate, but it's also dependent on the method that's used for analysis. And why is that? The mitochondrial DNA is circular and very condensed. You can compare it with a ball of wool. You see it on the right side. It's very tight, and close and condensed. And when you use the bisulfite pyrosequencing, that it can give rise to overestimating mitochondrial DNA methylation, also after BAM-HA restriction treatment, which should stretch the mitochondrial DNA. So because the bisulfite is, next slide. Okay, sorry, backwards one. Well, the bisulfate is protecting the methylated C, and if it can't be saved like that, you give an overestimation of the mitochondrial DNA. Next slide. And how can we overcome that? Well, we used an LCMS, SciX-AP, a triple quad, and we measured mitochondrial DNA methylation like a normal compound. We measured the C and the methylated C in the LCMS. We have described it in our recent paper. And the percentage methylation is calculated using a calibration curve. This method was according Zhao with optimized adaptations. We have looked into it carefully, and we could measure from 0 to 10% of methylation. Next slide, please. Well, here it is, a nice straight line. As a laboratory specialist, I'm very happy when I see an overall linearity from 0 to 10%. And we measured this in an independent mixture with varying amounts of MC and C. And we measured them in separate runs, each in triplicate, and the solid red line indicates the calculated values. Next slide. We measured also the myopathy patients in their cultured skin fibroblast. And we were a little bit disappointed to see that no overall differences in DNA methylation was seen. But we had an explanation for that. In pyrosequencing, we only have a specific part of the mitochondrial DNA investigated, the D loop and the side B. And in this one, we saw total DNA methylation. There's not mitochondrial DNA methylation and not specific the D loop or the side B. Next slide. But for every problem, there is a solution, and we are now in the run to use the mitochondrial DNA methylation profiling using the targeted fragmentation assay. And we isolate the mitochondrial DNA, we cut it in pieces, get it to some magnetic beads, and then the magnetic beads are taken out of the solution, is hydrolyzed, and then we can measure it via the mass spectrometer as well. So then we have the same, we hope that we can achieve the same results as we have seen earlier with the pyrosequencing. Next slide. To conclude with this presentation, we think that mitochondrial DNA methylation can be used as a next generation biomarker. Of course, it's a matter of debate whether it is there or it's not there, because there are not always conclusive results. And the overestimation of the percentage of methylation is dependent on the method of analysis. But when you use LCMS, you measure only the single analyte, the compound, and you have a clean measurement. Of course, we've tried some other material, and that is platelets and urine. The advantage to use platelets is that there is no nucleus in it. So we have solely the mitochondrial DNA, and urine is easily accessible, non-invasive, and that is for in the clinics a nice way to use. And perhaps we can use mitochondrial DNA methylation as a next generation biomarker for possible therapies. Next slide. This is our recent paper. It's 5 years work. I'm laughing a little bit because 5 years, it was very quickly done, I think. And if you like to have the entire literature list, then it is available on my upon request, of course, and you can mail me at this mail address keniezen9@gmail.com. Next slide. Thank you for your attention.
Yes, thank you, Dr. Niezen-Koning. We appreciate your presentation, and our audience appreciates it as well. There's one clarification question from our friend Alan Crowley that I would like to pose just for the understanding of our audience. Is it correct to say that as mitochondrial DNA methylation goes down, the capacity for muscle repair goes up?
No, I don't think that we can say that so well. No, I don't think so. That's the way to go. We have seen that the ATP generating capacity is decreased, but we haven't seen any aberrant velocities in measuring the capacity of the separate complexes. So we have one test in the clinics that can measure the malate oxidation, for instance. You can do that separately in a nice way. And well, that's the only thing we have performed. You can't say in vivo that there are changes in the muscle, because then you have to do some electron microscopy, and we haven't done that.
All right, thank you, Dr. Niezen-Koning. And we have linked to the paper that you cited here, the Mitochondrial Epigenome, an unexplored avenue to explain unexplained myopathies. So anyone who wants to access this paper can do so via this link. And now I would like to turn the floor over to Liz Parrish.
Can I ask a question? Do we get a chance for Q&A? Do we get a Q&A on her?
Yes, we do. So Bill, go ahead.
I didn't see an opportunity. I don't see how to say I want a question, but you had mentioned that the bisulfite treatment gave an overestimate of the number of methylations in mitochondrial DNA. Why is that?
Well, when you the bisulfate, I have a figure in.
Showed it how like the mitochondrial DNA is like a ball of yarn. So is genomic DNA in many senses. But I've never heard, if that's the reason, I would think that would underestimate the number of methylations, not overestimate. If that was the reason, the steric hindrance from the ball of yarn causing the bisulfite to be unable to get to the methylated cytosines that turn them into uracil. But what is the reason why you say that there's an overestimate of
you measure the cytosine. They are not. I have a picture of it, so I want to show it, but I can't find it at this moment.
Is it within your slide deck, or
no? It's an other slide deck I have. Sorry.
Okay. So I think we can arrange for this whenever you're ready.
I thought there was an easy answer.
Okay. Well, if you can find the picture, let us know, and we can elevate it later on during this salon. For now, let's go to Liz Parrish for her presentation, and we can return to the exchange between Bill Andrews and Dr. Niezen-Koning. So Liz, feel free to go ahead.
Okay, great. I'm probably going to back up these slides nearly to the beginning because we might as well do that. Thank you for having me. It's wonderful to be in such esteemed presence. So again, thanks. So my company is BioViva, and we're more of a translational engine. We obviously do science, and we're going to talk about that. The company was started in 2015 as a response to my son's diagnosis of type 1 diabetes in 2013. I had been basically volunteering in the regenerative medicine stem cell space for a couple of years and had heard of all this fantastic research that had been done. And when my son was diagnosed in 2013, I realized that, almost none of it actually translated to humans. It was all pretty much stuck in animal models, underfunded. And in 2014, I got lucky enough to meet Dr. Andrews, Bill Andrews, who's on the call here. And he's been an advisor to me ever since, and intrinsically involved in everything that I've done since the inception of this company. But let's get started because the most interesting thing is, what does this mean for you? So, basically, we want to take you where you want to go. We believe that the biggest limit to civilization evolving into something more, something better, solving bigger problems and going to bigger spaces, maybe outer space, is time. We are definitely limited by aging-related diseases. And as we proceed forward, how this relates to childhood disease, something that was my first initial drive to cure is that all of the genes that we look at can treat a childhood disease. And by treating the biggest medical unmet need, which is aging-associated non-communicable diseases, we can expedite those cures. And we can also, by scale, make things more affordable. So we target what's aging you. We look at the hallmarks of aging. There was a great paper that came out in 2013, just in time for me to get started. And it talked about, several things that are happening at the cellular level that give us some targets. Like, if you're going to treat aging, how are you going to do it? Like what is your initial target? I mean, Bill Andrews is spot on when he says, we should be able to know if we treated aging by looking at a person, but how do we get there, and how do we target these hallmarks? One of the biggest ones, I think, of the hallmarks of aging is telomere attrition, only in the sense that by lengthening telomeres, we seem to hit many of the hallmarks of aging. And most importantly, it's associated with lifespan. So we want health span, and I'm going to show you in this presentation how it affects health span. It definitely does. But if you want to break that bottleneck of lifespan, it just is a therapy that you're going to have to be able to do to make longer, healthier lives. And we need time. There's no sense in studying for 60 some years to just then get dementia and lose, your ability to function. So I call this Bill's gene. Well, this is actually Bill's enzyme. This is telomerase. He has done so much work behind understanding this gene and how its enzyme works on lengthening the caps of the chromosomes. It affects mitochondrial health. I think we'll show that in a paper that we produced that has recently been published. It helps with genomic stability. Then in that case, it could actually protect people against getting cancer in the future. It reduces cellular senescence, keeps those cells, healthier, and, maybe going through apoptosis rather than becoming, some old zombie cell. And it protects against biological aging. And so today, we're mostly going to talk about the work that we're doing in slowing the aging process. We are already moving forward into the work of reversing the aging process. And this gene will be very important in that. So the second gene we're looking at is something called Klotho. Klotho is a geroprotector. It's known to help assist in brain function, which is kind of cool. When mice were injected with Klotho, within an hour, they performed better on tests. Patients who, upon death, very unfortunate timing, who have been autopsied, who are upregulators of Klotho and had lots of beta amyloid plaques, basically like the symptoms of Alzheimer's disease, but still had really good cognitive functions, again, are upregulators of this protein. It's seen in people who are protected against things like cardiovascular disease and chronic kidney disease. And our interest in this gene is kind of the reason that we, for the time being, have dropped the interest in FGF21. Just in case somebody notices that that's not on our right now list. So follistatin is another gene. It is essentially a myostatin inhibitor. So myostatin is something that limits muscle growth, which is really great when you're young. You don't want to put all this energy and beefing up into all muscle, especially if resources are scarce. That's something that in the United States we don't have that problem right now, but there are still places in the world that do. And the problem with myostatin is that as we age, we get something called sarcopenia, frailty. We lose muscle mass over time, and we want to stop that. We don't want people to fall down, break their hip, lose their muscle mass. So this is a really interesting gene. We're going to talk more about this later in the presentation. We were initially interested in it for reducing frailty in an aging population, but there are a number of other things that it helps with metabolic disorder, and we'll talk about that in a moment. And then the last gene candidate that we're looking at closely right now just within this slide deck, not all of the genes that we're looking at, is called PGC1 alpha. And this is really interesting because it kind of speaks to the first half of this presentation. It's known to upregulate mitochondrial biogenesis. So more mitochondria, healthier mitochondria, and if the mitochondria are super dysfunctional, just essentially starting to force that cell into apoptosis. And so, again, this is a gene that's associated with exercise. You upregulate it when you exercise, but a lot of work is still being done on this gene. But it's actually quite exciting. It may protect against things like liver damage. And all 4 of these genes extended the lifespan of model organisms. So that's why they become candidates for us. Because when you extend health span with lifespan, that's really important. And our job is to get these to humans because that's what everybody wants. We need therapeutics for humans, not so much for mice. So we've been around for a while. We've been around since 2015. We started with AAV, which is a gene therapy delivery method. It's adeno-associated virus. It's a little fantastic beast. It has low immunogenicity, so it lacks the problems that they initially had with gene therapy. When it integrates, it is non-mutagenesis, or non-mutagenic. It doesn't cause cancer in humans. It has some problems. It has a limited payload size, but that was okay. That was a great place for us to start. We have 2 peer-reviewed papers around AAV, and we'll get into that real quick because I know we want to get to the newest paper. So aging-associated disorders, we actually got our first leg of the patent in this space. This is my data. This was the gene therapy I took in 2015 to start the company. Basically a cry out to the world that we are moving too slow. Millions and millions of people have died since I took that gene therapy, and vastly the world tried to treat it as a spectacle rather as something that was very serious, and that's a big failing because that leads to the death of millions of people. So this was essentially me calling out and saying we're moving way too slow. I will step up. I will take this gene therapy, and here's what we saw. We saw increase in muscle mass and decrease in intramuscular fat. I have had MRIs since the beginning until just recently because, we actually look at everything. We've had full body MRIs where we look for any mass or abnormality. There has not been any negative consequences to this therapy, but pharmacogenetics says that, somebody could have a problem. Probably most of the people wouldn't. Most people would benefit from this type of a therapy, especially in aging population. I had better metabolic health. Average telomere increased by 0.35 kb per year. This is probably a general good place to start in combinatorial gene therapies, and this is where we started. Better things are to come, and we can talk about that later. Then we went to dementia. We worked with a company that's offshore medical tourism. They wanted to do gene therapies. We wanted to assess data. We can't treat people, so we said, please let us assess your data. And these were 5 patients who took, again, AAV, adeno-associated virus gene therapy. And they took both telomerase reverse transcriptase and clotho. We saw improved cognitive scores. That was something that was pretty, really amazing, actually. And we saw an increase in their shortest telomeres, which we didn't expect to see with such a small dose of telomerase reverse transcriptase gene therapy. What that means to anyone watching is maybe in smaller doses, we can have an effect on bodies. And again, that's Bill Andrews' area. And there is getting the wheat and the chaff separated and, making sure that they just didn't have hematopoietic stem cell turnover or whatever, and just didn't have like fresher, better telomeres from something else. So let's see. So here's where we're going. So what we learned in the past is AAV is fantastic delivery method, except for when it's not. Not really great in super high doses. There are certain things that you have to do to ensure that patients don't have an immune response. And it actually has a really small carrying capacity. And that really limits us, because we want to create combinatorial gene therapies. We want to deliver precision medicine, and we want to be able to deliver several genes associated with longer and healthier lifespans. But if a patient has, let's say, an outlying problem in which those genes may not be beneficial in total, they need something else, we would be able to add that. This still has a limited size, but it's much bigger than AAV. And so this is a paper that was just published in PNAS just probably last week or the week before that. And we're really excited about that. I don't have a proper slide around it, but I'm going to show you what happened. So in this paper, we're essentially trying to find a new gene therapy delivery method. The reason that we chose two genes, telomerase reverse transcriptase and follistatin, is because they had a known effect in mice. So when you're delivering a gene therapy, you're actually delivering a couple of drugs. One is the delivery method, and one is the therapeutic gene, right? So you have to test both of them. So you have to have a control that has nothing. You have to have another whole group that just has the viral vector. Then you have to have some way to measure whether the vector delivered genes. And so our company tends to choose genes that have already like a really good meta-analysis. They basically, different labs have already used these genes and kind of know the consequences of them. And that's why we think that they can be expedited to human use faster than an unknown candidate. And so we used telomerase reverse transcriptase and follistatin. And we decided to just go ahead and do a lifespan study whilst we looked for endpoints around treatable conditions that the FDA might be encouraged and so inclined to allow you to use these type of therapeutics. So what we saw is telomerase reverse transcriptase increased the lifespan of mice by 41.4%. And this was pretty exciting because remember, we're looking really to create a better gene therapy delivery method. And then the mice just keep living. And I think that probably every researcher probably has that happen. For me, this was a completely new experience where every week you're just literally on the edge of your seat. And this was one of those moments. And the follistatin mice lived 32.5% longer, which also was fantastic. And none of the mice treated with TERT or follistatin died within the range of the untreated controls, or the treated mice that only had the vector, the cytomegalovirus by itself. So that was really cool. So they were all outside of that range. So the TERT mice maintain their body mass longer. We'll look at a graph on that. And the follistatin mice increased their body mass. They were big girls. And they were pretty active on the bottom of the cage. The glucose tolerance was better in both. I just have to tell you, telomerase reverse transcriptase performed better in every single test than follistatin, but follistatin really held up pretty darn well. The glycated hemoglobin, again, best in the TERT mice, but closely followed by follistatin, which basically tells us they're probably both really good candidates for metabolic disorder. And task performance, the TERT mice were just faster at everything. The follistatin mice were heavier. They were super active on the bottom of the cage, not so much excited about jumping and stuff like that as the TERT mice were. So when this ties into the first presentation, mitochondrial health, this was we really wanted to look at this. We wanted to see what was happening with the mitochondria of the mice who were treated compared to young mice. And in both cases, these mice performed better. Of course, telomerase reverse transcriptase was outperformed follistatin in mitochondrial health. And here what you're seeing on the right to the two right far corners is the TERT and the follistatin mice. And then on the first column down is the young mice. And they have a very similar mitochondrial appearance in cristae. And you can further look at the graphs on the right for the performance of these mitochondria. This really points to better mitochondrial health. We were really excited about this. You can definitely look at this in the paper. On your screen, this is probably too small to understand most of it, but please go to PNAS and look at our paper. One of the strongest proponents of this paper that's really cool is we did both intraperitoneal delivery, straight into the mouse, and we did intranasal. And they both performed really well, about the same. And that's kind of like taking gene therapy to a new step. So, like snorting your gene therapy for a longer lifespan. And we did redosing. And so we're hoping that this sort of redosing of gene therapy will translate to humans, to immune system response. On this slide, you're seeing what happened with body weight on the top middle. On the top left, you're seeing our gorgeous mice. You're seeing how fabulous the follistatin mice looked compared to the TERT. The wild type mice, not so great. They lost their fur. Our treated mice definitely maintained their fur longer, and the TERT mice maintained their appearance actually for a very long time. Actually, some of the vet technicians thought that they were 8-month-old mice after they were treated. And these mice were treated at a later stage in their life, about the age of 56 of humans, which is really translational to when we think that people probably right now would be good candidates for gene therapy. Right now, it's really like more the terminally ill. And we're going to talk about a paper that Bill and I are putting together for that, for expedited use of those type of technologies later. But for all intensive purposes, these will be moved back to younger and younger ages. And probably in the future, people much younger than 56 to 60 years old would take gene therapies to not accumulate the damage and of the aging response in the future. So, I think they'll be used more like immunizations. But anyway, you can see here that body mass matters, and especially in aging animals. And I think, Bill, that was something that you told me a long time ago. You said you've never seen a fat, old cat. And that's the case in most animal models. They get thin, they get frail, and they get pretty emaciated as aging happens. And so maintaining body mass is important. And that is what we saw. Here we also see blood glucose levels and the beneficial effects of this therapy across the board. So, our goal is to hope that this translates to humans. And create, longer, healthier lived humans. Here, really quick, we're going to look at telomeres. This was, of course, also the cytomegalovirus gene therapy. So there's a couple of different things going on here. C and D are essentially all of the mice in the study, a comparison. So the telomerase reverse transcriptase mice have longer telomeres. The box in E that you see on the bottom right, this is when we compared them to young mice. So this is, you can see that the telomeres of the treated mice are shorter than the really young mice. but they're still longer than the follistatin mice and the controls. So that's pretty cool. That was a lot of fun to see. And then, I mean, when people are watching, like if you want to know the molecular mechanisms, what's happening, what we know, what we don't really know, especially in the space of telomerase, please ask Bill Andrews. All of the mechanisms that sits with our scientists. That's not something that I should be speaking on. What I really am speaking on is the translation of these technologies to humans and how they might affect humans in a positive way, and and what really matters to the consumer and adding value to human health. And I really like this slide because I feel like it kind of it's the best summary that we could come up with to understand that. So on the top, you see mice, and this is mouse lifespan. And on the bottom, you see humans. And so the top is in months because humans don't live as long. I mean, mice don't live as long as humans, right? So that's why you do studies in mice, and the humans are on the bottom. And so if our mice, let's say that their survival curve of the wild type mice laid in just under 80 years old, about when most humans die these days. People are living longer, but this is kind of like an average lifespan. It's actually gone down since COVID in the United States, but okay, let's just say that this is an average lifespan. You can see with follistatin, we're getting you past 100. So just this one gene therapy, if it translated directly from mice to humans, we're looking at a longer and healthier lifespan. I mean, these mice were healthier and more functional, and that's really what's important to us, right? We don't want to live, old and sick for a long time. And the TERT, the telomerase reverse transcriptase mice, if that was translated to humans, that's over 110 years. And so, this is really exciting. And then finally, so what we're doing now, if anyone's interested, you're probably just like, okay, give me a drug. Well, it takes a little bit of time. So we're working on human cell data now. We're working with a nonprofit to raise money for BioViva to redo the dementia study with more patients and a higher dose in a way that will be IRB approved, in which we hope that the US FDA recognizes it for an RMAT position, accelerated use for humans. We're applying for grants in the area of space. Actually, the CMV gene therapy for mice right now is being used at a university. Can't talk about it too much yet, to test against radiation exposure. So that's pretty exciting. Might help people go farther into space in the future. We're expanding into the combination therapies with the human cell data. So now, it really is the time to ramp up and start to bring together and see if we can get like an exponential super wave of benefit with multiple genes. Our company will be going into a Series A, hopefully later this summer, to start position ourselves for clinical trials. And of course, everybody's asking, are you interested in partial cell programming? Of course we are. We are interested in treating aging, and so you'll see some data come out around that in the next year or two. I mean, it takes a while. We always like to offer something free to the world at large. We're really huge education engine along with our research and development. And so if you're interested in gene therapies around some disease that you might be suffering with, or you're a researcher and you want to use a database to study a specific disease or a specific gene, go to bioviva-codekeeper.com. We are open sourcing the cure, we call it. We are trying to make it so gene therapy is easy to search. You can find out what's happening out there, and you can better understand the bottlenecks. For instance, a lot of this technology sits in murine models, meaning mouse models, because it really doesn't have the funding to go anywhere else. And so just because you see something in the news doesn't mean that those researchers were able to bring it to clinical trials. And it's often not for lack of good data, because usually if something makes a paper, it's because it had good data. We really have to start publishing bad data too, so that we can know what not to do. But anyway, these papers are vastly all of the gene therapy papers that are out that, show, positive effects in one place or another. And so, I really encourage people to get out there and start funding this technology, especially if a disease runs in your family. Go find out what's happening. And if you don't find the paper, submit the paper, and we'll have a PhD student parse through it and make sure that it's uploaded properly. So that's essentially what it looks like. You would go, you search, you get to look at papers, and you get to submit papers. And we're hoping that this expedites the use, development, and the funding around gene therapies of a variety of biotechnology companies that can really be game changers for the future. I'm not going to show the next slide because it has a limited number of our team, and it actually doesn't have all of the team members on there that actually make this happen. And so I think that that's not right. This presentation wasn't really ready for prime time. A lot goes into this technology, including everyone who came before it to get us to the point where it happens. And our goal is to really finish that off and translate it to humans. We don't think that we work too fast. We think that vastly everything is moving way too slow. And for, over 100,000 people today, it was too slow. And tomorrow and every day until we get these drugs into humans. And so lastly, I just wanted to end with that Bill and I have a paper that we are getting published that is around what we call best choice medicine. And it is a new regulatory path for the US government, not the FDA, the US government to consider, in which would decouple early use of drugs away from the US FDA into a path that would then be connected to them upon good drug candidates. Because today's Right to Try Act and other moves towards early access just do not actually speak to the drugs that we're talking about today, these more precise genetic therapies that haven't been in early phase trial, but desperately patients need them. So that's it. And then, of course, Bill, if you have anything to say, please do. But thanks for having me.
Yes, thank you, Liz. We greatly appreciate your remarks and your presence today. And Bill, please go ahead if you have any additional comments to supplement Liz's presentation and discuss any of the topics that have been raised.
I just want to mention that George Church is one of her scientific advisors, and that you would have seen that on that slide she just didn't show, which is she's got a lot of support and background from everything she's doing. Yeah, I'm available for questions if Liz can't answer it, though I do consider Liz somebody who understands the science better than most scientists in the field. So I'd be surprised if she can't answer them too.
I have a few questions for Liz and for Dr. Bill. So there was a recent study on glycine and acetylcysteine. They were not general antioxidants, but just precursors. And what I'm hinting at, so you have the gene therapy going on, the telomerase, maybe the alpha clodo. Are there certain precursors that can be added that will enhance the effect of the gene therapy even further? And the second question is, the longest lived mice species ever on the planet are the Ames dwarf mice, who have problems with growth hormone not problems, but altered growth hormone signaling, and that's why they're small, but they live exceptionally long. So are you guys looking at growth hormone type, so altering the growth hormone, exactly, not activating growth hormone, but really trying to inhibit growth hormone receptor signaling to extend lifespan, as that has been the longest lived mice we've ever seen.
Can you ask, what was the first question?
Gosh, yeah.
Also, the first question is, since we were talking about things like telomerase and follistatin.
So there's two types of telomere shortening. One is just the simple result of when a cell divides and makes a new DNA. The new DNA is made shorter. There's no such thing as degradation or unraveling or anything like that, but terms people often use. Telomere shortening is mostly caused by just the cell's inability to make telomeres longer. But there is something called accelerated telomere shortening, which is caused by free radicals, inflammation. Well, inflammation is more actually due to making cells divide faster, and so telomeres get short faster. But the antioxidant or free radicals and other harmful agents can cause accelerated telomere shortening. And like something like N-acetylcysteine, which becomes glutathione when it gets inside the cell, actually will help decrease the rate of accelerated telomere shortening. But in humans, we already have 10 times the level of antioxidants that mice do. So it's not going to play as big a role as it does in mice. But there are a lot of products available right now where anything that contains an antioxidant or anti-inflammatory is now being hyped as something that will help keep your telomeres long, because it decreases the rate of accelerated telomere shortening.
I wasn't meaning that precise. I was meaning the precursors to telomeres themselves. So you have some nasal telomere gene therapy. So what are the precursors to lengthening the telomeres? That was where I was aiming at.
Can you tell me what you mean by precursors to telomeres?
So what are telomeres? So they're just the DNA base. Is there any way that the telomere gene therapy can be made even more powerful by just adding certain things besides like radical things, but certain supplements?
Are you talking about substrates when you say precursors, like
Substrates.
nucleotides? I just never heard that anybody use the term precursor of a telomere. But yeah, in order for telomerase to work, it does require, and it does benefit from a lot of the proteins that do bind the telomeres opening up, like TRF1, TRF2, a whole bunch of telomere binding proteins. But telomerase, when you overexpress it, seems to work so well in lengthening telomeres. If you can get telomerase into the cells, it lengthens the telomeres quite effectively if you put enough telomerase into the cells. And so I don't know if there's any advantage to actually figuring out a way to actually increase it longer, unless you have a telomerase inducer that's really weak. And in the case of Liz's study, her telomerase isn't really weak. So they get longer. Why get them longer faster? I don't know. But there are studies where people are looking at inhibitors of some of the telomere binding proteins to try to open it up so telomerase has more access. But they're mostly looking at mouse cells that already have telomerase produced in all their cells. And so that offers them a benefit. But since human cells have no telomerase, except for our primordial germ cells, it's not going to really benefit humans at all until somebody starts producing telomeres inside of a cell.
Thank you so much.
Yes, thank you very much, Bill, for
You had a
answering
second
Simon's questions.
You had a second question, which I can't remember what that is now.
Oh, so the second question is, if we just look at long-lived mice models, the record is the growth hormone signaling altered a mice model called the Ames dwarf mice. Ames dwarf mice live approximately 50% longer, and the ones that are the Ames dwarf mice that are then put on caloric restriction that just broke every single record that I've seen up to this point. And I was thinking, like, okay, you guys have something going on with growth hormone signaling to see if since that's the record holder of lifespan at this point in time. Thank you.
Bill, I mean, we did look at those studies, like many years ago, and those are knockout, like from birth, right? So, I mean, like, so human development, like most of us are beyond that, and I'm not right now, like in dwarfed humans, I don't think that we see longer lifespans, but I'd rather have Bill answer that. We're definitely not looking at that at this point, because, like, you know, most of the population is beyond development. We don't really get into embryonic editing or, you know, pre-development type therapies at this point. So, I mean, like when I think of something like that, like still there's, you know, regenerative capacities to human growth hormones. So like, you know, maybe what is done in the past in certain studies really isn't like a model for future humans. But I'm like totally guessing, because that's not my area. But Bill, do you have any answer to that?
Well, yeah, I'm one of the inventors of HGH. I do know a lot about HGH. Back in the late 70s, early 80s, I was very involved in the Genentech and stuff when we were developing HGH. But it's going to be more effective in mice than humans. Probably the best study underway right now to look at HGH in humans is being done by Greg Fay, and looking at the thymus regrowth when he's applying HGH along with some other things, metformin and what's the other one? Something I can't remember. Quercetin, or I can't remember what it is, but DHEA. So DHEA, metformin, and HGH are having a very good effect on rejuvenating the thymus in humans. But I don't know. So the Ames dwarf mouse, I mean, that would be worth looking at. But and so would the naked mole rat, who is also a very, very long-living, unengineered animal. But the problem is that, and most people don't know this, is that when you take mouse cells and put telomerase into it at a very high level and lengthen telomeres, it has no effect on delaying senescence at all. Okay, so it's really unclear as to how telomerase and telomeres are playing a role in mouse aging. Clearly, with Maria Blasco's studies and now Liz Parrish's studies, telomerase does increase the lifespan of mice and health span of mice somehow. I don't know if it's the moonlighting activity that some people think that telomerase has, which I'm sure it does have moonlighting activity, or somehow the treatment is inducing like naive immune cells to suddenly start dividing because their immune system is reacting against the treatment, and that's just providing more cells that have longer telomeres. So, but it's really hard to say what's going on, especially comparing mouse to humans. We find everything we do, especially when it comes to telomere biology, just doesn't seem to behave the same when we compare mice to humans.
Okay, thank you so much. Thank you for the elucidation of the topics.
Yes, thank you, Bill, and thank you to Simon as well. And Simon, I believe we have the picture that Dr. Niezen-Koning wanted to show in response to Bill's earlier questions. So let's take a look at that. And Dr. Niezen-Koning, if you would like to explain this diagram, please go ahead.
Yeah, thank you. I think this diagram is familiar with a lot of people. The biosulfate sequencing relies on the conversion of every single unmethylated cytosine residue to uracil. And then if the conversion is incomplete, the subsequent analysis via PCR amplification will incorrectly will be incorrect, and then you get an overestimation of the cytosines. Is it?
Why does it? Well, why wouldn't it? So Is there something preventing the bisulfite from turning the cytosines into uracils, or actually overdoing it? Or yeah, no, it'd be underestimated. So it'd be interfering with bisulfate to turn cytosines into uracil, therefore giving an overestimate. But why?
Yeah.
I mean,
Yeah.
we do those things all the time here on genomic DNA.
Yeah, but genomic DNA is more loose, and mitochondrial DNA is more condensed, as I said in my talk. And that's why we think, and people have written down in literature as well, that due to the condensedness of the mitochondrial DNA, the bisulfite can't can't convert the unmethylated cytosines, and that's why they are not. Then they are not converted, and there's an overestimation of cytosine. And
So-
that's only with mitochondrial DNA. It's not with genomic DNA, because that's quite a different case, and that is, sorry to say, but it's more straightforward.
Well, they're both condensed inside the cell. Are you saying that the mitochondria DNA is really condensed when you extract it from the mitochondria?
Yeah, when you isolate it from mitochondria, it's very condensed. Yeah, it's like wool.
I'm just thinking of our gel electrophoresis when we run supercoiled DNAs, even mitochondria DNA. We don't see it being any more condensed than like a regular bacteria plasmid. If that's the explanation, I'd like to learn more about that, because.
Okay, well, we can have a talk afterwards, perhaps, when or via mail or something like that, if you like. That's good.
I can absolutely connect the two of you via email, and indeed, this is one of the consequences of our virtual Enlightenment salons. We get to host conversations at the cutting edge of scientific discourse and then get the subject matter experts connected to one another. So I think this is excellent. And speaking of connections, we have a policy connection here with Dr. Edward Hudgens, who has been working on FDA reform and advocating free to choose medicine for a long time. And we have had a prior virtual Enlightenment salon with him on this, as well as various conversations in other virtual Enlightenment salons. And Liz mentioned a proposal that she and Bill are working on to also provide a pathway for patients to test these early stage gene therapies. So Edward, you have some questions and comments in this regard, so please go ahead.
Oh, yeah, thanks. I'm Ed Hudgens, and I think I know most of you folks here, by the way. Excellent book for those of you who haven't read it yet. I've been working for quite a few years on FDA reform, and there's a thing called free to choose medicine that I've written a lot about. There's a book about it, and right now there's legislation before Congress. It's called the Promising Pathways Act, and what it is, a version of free to choose medicine. It says basically that it's modeled after an AIDS reform in 1992 that allowed AIDS patients to secure medicines years before they were available. Because if you're an AIDS patient and you're told, well, maybe in 3 or 4 years we might have something and you're going to die in a year. Well, that's rather cold comfort. What this does is it basically sets up provisional approval or a parallel track that if a certain kind of medications under certain restrictions have passed a phase one efficacy tests, and at least one phase 2 or phase 1 safety test, at least one phase 2 efficacy trial, the sponsor can offer them on a parallel track, and patients can choose to try that medication, which, remember, are being used in clinical trials right now anyway. So it's not like it's a dangerous medicine. But the secret is that their experience will have to be put into a database with their privacy protected, of course, so that researchers can look at this real world data that's flowing in to concerning whatever the particular medication is. What I've tried to do, and I literally have a meeting about this on Monday because there's been pushback on Capitol Hill about this. I inserted into that legislation that it should cover a number of things, including the processes that constitute aging. A lot of congressmen were uncomfortable with saying aging. What the heck does that mean? Well, what about the processes that constitute aging, such as telomere shortening, mitochondrial issues we just discussed, et cetera, et cetera. And so I put that into the legislation. And as of like Thursday or Friday, when I met with a guy who had been working the Hill on this, he said, well, a lot of them want to have that taken out. But the point is that there's literally action right now to do this. So Liz and Bill, I'm very interested in what you guys are trying to do, because I've worked this area in public policy for quite a few years. And so I think there's an opportunity for us to talk and see what you guys are doing, how it might fit in with some things that I've been doing for the last couple of years. Because the idea is to, getting aging declared as a disease is going to be tough. But if you can basically at least say it's a treatable condition, like Aubrey de Grey likes to say, or the processes that constitute aging, the FDA has to look at those, as opposed to say, well, it doesn't cure, it's not trying to cure a particular disease, so we can't even look at it. So anyway, that's what I'm doing, and I'm very interested in talking with you guys. And maybe I'll leave with a question, which in a sense you're already working on. If you could reform FDA right now, what exactly would you do?
Liz, can I just go first really quick? I talked to Donald Fink quite a lot about the definition of aging and aging a disease and stuff. And I agree with him 100%. Aging isn't measurable. Okay, there's no way to measure it. We have all these biomarkers right now that we can measure, but none of them have actually truly been shown to affect aging. They just correlate with aging.
Yeah.
And so we can't measure aging. So the only way to really go and you can't really define it as a disease until we cure it. That's why I always say cure aging. My book you just showed is curing aging, but we can't call it a cure until after we cure it, and then everybody will agree it's a disease. But until then, we can't measure it, so we don't really know. So focusing on diseases of aging is something that we have to do. And I don't really participate myself on trying to get aging classified as a disease, even though I know it benefits everybody. I just think it's going to be pretty impossible to get it classified as a disease. Even though I like to call it a disease, I'm a first person who'll say it really isn't. We can't classify it as a disease yet until we actually cure it.
Right. Yeah. And that's why I'm not pushing for that, because again, there's all these questions that opens. Whereas if we say something like processes and, that constitute aging, then you can talk about telomeres, you can talk about telomerase or whatever it is.
Yeah. Liz, do you want to go ahead?
Yeah, sure. I think, that is kind of interesting for me, because, I definitely consider aging a disease. I think that it needs to be accounted for as such. As Bill said, when something is definitively treatable, then I imagine those classifications would change. And so oddly, what you'll find with our paper is we are not really pushing for aging to be classified as a disease. It's essentially this paper is, and what we want to have happen is a lot more about streamlining the use of drugs. And so, for instance, with right to try legislation, if something is in phase 1, or especially if it's in phase 2, people can get access to it, but companies don't necessarily need to report on the effects of the drug when they use it outside of a clinical trial. Expanded access also gives the rights for physicians to use some number of drugs and report minimally on them, but they have to do a lot of paperwork in order to use them. And so the patient often dies before they get access to them. So what I am saying and what people are supporting is that actually newer drugs, so drugs that you take right now that are regulated are old drugs. Especially at the rate of development. Genetic therapies are more precise, but this paper doesn't hinge just on genetic therapies. It hinges on the better known relation of these newer class of drugs, gene and cell technologies.
Mhm.
So we're actually saying that a drug in a clinical trial isn't necessarily a safe drug. So, you kind of implied like there are these drugs that have some amount of safety and efficacy, but pharmacogenetics would say that actually they're not safe for all people. But they probably are safe for most people once they've passed through regulation. So even when a drug is regulated, it's not necessarily a safe drug for you. So what I am advocating for is that now that we have more and precise medicine than we did in the past, especially with the way the FDA was basically developed around some of the drug failures and problems that we had with small molecule drugs, that people should get access before drugs are in phase 1 for terminal illnesses or illnesses that have no treatment. So this is definitely a radical shift, and we're not actually trying to advocate that the US FDA does this, that there is merely a different track in which can work in parallel for successful drugs. So giving people in the United States, starting here or any country, the dignity to try a drug who have a terminal condition, who have no other outlet to drug development that would help them through the regulatory body. They should be able to take these newer and more precise medications to be part of essentially bringing the human data to the table as to whether these drugs will work to improve their condition. And that sounds like a radical shift, but again, it's not. Once you realize that clinical trials are not safe, they're no more safer than just taking a drug with an unknown effect, honestly. Even a regulated drug comes with a lot of risk. But these drugs are a totally new class of drugs. And so this doesn't suit my company's gene therapies. It suits a number of emerging biotechs. As a matter of fact, most biotechs, their technology is far in advance to anything that you could ever get access to today outside of this type of use. And so it's essentially, the paper is using what we have learned from medical tourism, the ease of access, the desire for people who want to participate in an altruistic event and new technology because there is no other treatment for their condition, and the dignity in order to give people the right over their own human body to access this type of technology. It only sounds radical when you try to shorten what it really is. It is actually common sense use of technology that may help people now. And so if there's any way in which you want to be a part of that or assist with that, please let us know. We think that, this is detrimental. And, for instance, the US FDA has, they have their own caveats. They're held responsible for when a drug doesn't work very well. And so there's a risk aversion in the whole drug discovery process. And yet, drugs go into clinical trials that are not safe. And there have been regulated drugs, 10 really dangerous regulated drugs that hurt a lot of people. So, we have to put aside that we think that somebody can do a perfect job, and we need to essentially help the people who work for the US FDA find better candidates by already having a drug profile, a human profile on a drug before their career hangs in the balance of approving that drug for a clinical trial. So it kind of like would help everyone.
Just to finish up, we should talk offline about this, because a lot of what I'm trying to do is exactly along those lines.
I can tell. I can tell.
The people I'm working with, by the way, a number of the leaders, in fact, the one I met with on Thursday after he finished his rounds on Capitol Hill, had his daughter die at, I think, 8 years old because there was a medication that was being tested. It was in, I think, phase 2, couldn't get access to it. His daughter died. You can imagine how frustrating that is. And he's got a whole network of people who've had that happen. Also, in terms of the safety, as you know, there's a whole FDA system for monitoring, drugs post-market and for updating labels, meaning, now we know that it's not good for this, but it's better for that, and so forth. So even after it's been approved, there's a whole system of collecting data. So anyway, yeah, we should talk offline because I'm literally working on these kinds of
So
issues.
let me, before we go offline, let me just add that I've been involved in 7 clinical studies now, and almost every time when I would sit in our clinical affairs meetings at the companies I'd be working with, and I'd be hearing about what we're actually going to do, I would say things to the effect, we can't do that. That's going to hurt the patient. That's going to put the patient at great risk. And the clinical affairs people, so it's not just the FDA that's a problem, it's also the companies. The clinical affairs people would say, yes, we can. The FDA gave us approval. So since they gave us approval to do this, we can do it. But I'll tell you, I've been very shocked at, I do like a lot of members of the FDA. I've been involved with Philippe Sierra and Donald Fink and stuff like that. But I find that a lot of times when people try to get FDA approval, the FDA will approve it even when it's not safe. And a classic example is like the bubble boy disease with gene therapy. Any beginning 101 molecular biologist would have said that if you put a vector that integrates into the DNA randomly into a person, that's going to give the person cancer. The FDA approved it anyway, and yes, 5 out of 7 boys got cancer as a result of the vector integrating into the chromosome. So I don't think the FDA can officially be somebody who can be considered qualified to be overseeing safety. I think that safety has to be in the hands of the principal investigator.
I think, Bill's point is, and you see that in some of the EMA therapies where, things can move a little bit more quickly, but they're on the backs of those who have the principal investigators, the companies that are behind the technology. And when you have a great responsibility like that, you really need those people to be taking the chance, and you also need the public to understand the risk. So I think that, what Bill's saying, and that's why he was so vital to the paper, is that, when you don't hand a responsibility to somebody who doesn't know, and you hand it down to the people who are actually in the trenches using it, developing it, then things are done at a far different level, in which it indemnifies those folks who don't want to be part of that, and puts the responsibility, a heavy responsibility, but a vital responsibility, on the developers of a drug.
I have one last really small remark on the legislation part. So this is practice versus theoretical ivory tower. So we do know that aging is correlated with 90% of human diseases. So an indirect route that me and Dr. Niezen-Koning have been trying, although we've never been able to organize it, was an aging cohort study, really specific on aging in either a Caribbean island or in Colombia. I mean, we haven't got that from the ground yet. So basically, there are many things you can do. You can show like an anti-aging therapy, such as a gene therapy, or be it next generation drugs or combination therapies, or things like fasting reduce your odds for diabetes, heart disease, strokes. And as a side, so you're really showing, okay, we can reduce diabetes and heart attacks and cancer, or one of those. And as a side effect, you also reduce aging. And if you can show, hey, I'm reducing disease, and as a side effect, these people have longer health span, in practice, that works quite beautifully as well. Although an anti-aging, really population level, I'm talking here about a few thousand people cohort study on anti-aging hasn't been done yet. Although they're trying in Singapore with some technology there, but we were trying to organize something in Colombia and in the Caribbean island on Aruba, really large scale, but it hasn't gotten off the ground yet.
Yeah, I think that, like, when you're looking at like human lifespan, the problem is like humans are so long lived, and so even though I believe that aging is a disease, I believe that it will be treated as a disease, hopefully in the near future. We still have to look at endpoints that could allude to that a human would live longer. And so we would choose, the symptoms of aging as a disease, which is, less complex than the whole. So we would pick a part, and we would pick, like you're saying, something like metabolic disorder, or type 2 diabetes, sarcopenia, pulmonary fibrosis, something in that space. And then we would try to prove that we could affect that, rather than trying to get a larger cohort and then having all the rest of the population die. But obviously, the point is to get a drug approved so that people could then maybe use it off-label for other conditions as well. So you're trying to find a low-hanging fruit and trying to find funding to actually target one of these low-hanging fruits in order to get through and do that. So I commend you. I don't think that we can wait for most people in a study that would be relatively healthy to be treated with regenerative medicine to then go on to live substantially longer than other people. I mean, we kind of already are doing that, but it's not helping the biggest stakeholders in this process, which is the 100,000 people who will die today. And aging associated non-communicable diseases are the biggest killers now on the planet. Non-communicable diseases are responsible for 74% of the deaths globally, and the 4 top killers are aging associated in the non-communicable space. So yeah, this is, we're all going in the right direction, and it's going to take many heads and many people working in the right direction and supporting each other. And, like how I always like to end a conversation is, make sure that you're being part of the solution for everyone. Don't be a longevity troll. Don't go, oh, these people shouldn't be researching that. You'll never hear me saying that, because we need everyone in, all hands on deck. Support each other, make it positive, find out how you can work together or not, share your research. We've done an extraordinary job for an actual company to actually, share research instead of keeping it to ourselves, trying to open that code in the gene therapy space and, continue to do that in the area that you're working in. But, if you don't like something, unless you're an expert on it, talk to the company and try to get more answers. And, it's really crucial at this point. We are so close. Don't take a side, play all teams, because we're getting there. With the partial reprogramming, the telomere lengthening, the mitochondrial dysfunction being addressed by different therapies, endpoints, the glycation endpoints starting to be addressed. I mean, you may actually be pretty excited in the next 5 or 10 years.
By the way, I'll just end my point. Simon's point is right on the mark, because the memos I've been sending back and forth in the last couple of weeks have basically pointed out that here are the diseases that are so prominent, are prevalent in people who are older. And so if you want to talk about, heading off that whole list, then these kinds of therapies, and if you don't want to call it anti-aging, well, these are ways to head off the, before you get cancer, Alzheimer's, and all these other things. I'm using exactly the argument that Simon put forward there.
Excellent. So thank you for this quite enlightening policy conversation. Now, we have a number of questions from our audience that I would like to address in the next 21 minutes that we have. First, this is an issue that Bill touched on briefly. Eugene D. wonders, do the BioViva human studies provide useful information for the aging science community to agree upon one or more human biomarkers of aging? And as I understand, your outlook, Bill, is that searching for specific biomarkers is a bit futile, because we essentially know what aging is empirically, and the key is to reverse it in some way that would be obvious to everyone who is looking at the person in question. Is that a correct understanding?
Yes, kind of. And like, let me just address Simon's comment too, because you can have the one. I consult for a lot of companies right now for years that are setting up hospital clinical studies in Colombia, Cambodia, other places like that, with trying to treat an aging-related disease, hoping that a side effect will be that they see aging itself is treated. But how do they know that? That's the big question. And there's no way to measure aging. The biomarkers are all correlated with aging. There's people saying that now even methylation, you can actually have biological epigenetic markers, but I'm not sold on any of the studies that I've seen that's showing that they're really biological aging. It's just still chronological aging, and maybe some health things are getting reversed. But I think the only way we're ever going to know if a drug actually treats aging, and when I say aging, I'm talking about health and life, is to either one, wait 50 years to see if the people actually live longer and healthier compared to a control group, and two is to reverse it. Okay, I don't think, in fact, what it comes down to, I don't think we're ever going to really be able to show that we've come up with any treatment for aging until we find something that can reverse it, because everybody can tell the difference between somebody like Betty White, if she was still alive, when she's 95 versus 25. Okay, so if we could actually do that, I call that, I don't call that empirical, I call it qualitatively quantitative, QQ. We work with QQ scores here all the time. We can't measure it, but we can definitely show photographs of two different people, same person, different ages, and everybody can tell which photo was taken first. Okay, so we do have that kind of measurement, but we're only going to know anything about whether or not anything is actually treating aging until we see it actually reversed to the point of where a human looks, feels, and behaves younger. And so the terms of the biomarkers, yeah, I would say, yeah, the BioViva study was really, really good, and they used several biomarkers to measure it, but I don't think any of them I don't think the study can say that any of the biomarkers were really any better than anything else in terms of measuring aging. I can tell you this much, even though people always say telomeres are my favorite, I can tell you telomeres are the hardest things to measure there is, and most people cannot do it correctly. So if I wanted to really choose something right now to measure my aging, I would choose DNA methylation or IgG glycosylation. I think both of those are a lot easier to measure and can give you a more consistent answer, and they're correlated just as well as telomeres are.
So I had a small thought, a really short thought here, is why don't we, so this is Enlightenment Salon, we're having enlightened ideas. How about something that Dr. Aubrey de Grey alluded to in his classic ideas, the Methuselah mouse idea. So I have, even with the genetically engineered or mice that are being thrown extreme amounts of certain compounds or etc., even so, there's never been an immortal mice. I have never seen a mice that's lived forever, or at least that keeps living on. And if we're really trying to set the precedent here, and with combination therapies, and like Liz said, looking at all hands on decks, looking at all techniques, be it like augmented fasting, gene therapies, next generation drugs, or be it whatever, really thinking of a combination of things that can basically make a mouse keep living really, really long, so that something like that. So making an immortal mouse study might be something like that, if that's even possible. It's never been possible, but we're pioneers, so we can try something along that line.
What's always been possible, it's just never been successful. Okay, lots of labs are right now trying to create the immortal mouse, but we're finding that mice age by entirely different mechanisms than humans. Okay, there's a lot of studies you can do on, even in the petri dish, with looking at aging in human cells, and they're completely different when you do the same study in mouse cells, and vice versa. So it's like, it's been true for like every cancer drug that I've been involved in developing worked beautifully in mice, but then failed miserably in humans. It's like mice are not a good animal model for studying drugs for humans, but they're good for safety studies. Okay, they're good for safety, but not necessarily efficacy. So I still encourage people to do safety studies with mice. But I think that for a lot of my presentations, I've said that we ought to be switching to something more human. And I think the best animal I found so far is the Pygmy Marmoset. Pygmy Marmoset is closely related to humans, but it turns out not all primates are. The Madame Bertha's mouse lemur, which people think are close to humans don't age anything like humans. They're more like actual mice. Mice seems to age by oxidative stress and mitochondrial dysfunction, but they have a whole bunch of things programmed in them to end their lives early. And from an evolutionary perspective, there's a great advantage to survival of the species by eliminating the old. Okay, that's something that is hard for a lot of people to understand, because evolution, why haven't we already evolved immortality? Well, it turns out the survival of the species in a rapidly changing environment is going to be more successful by knocking off the old, because it encourages the young to interbreed, to shuffle genes more to increase the chances that somebody survives a rapidly changing environment. So it's like mice are totally different than humans. It's mice evolved an aging process that has nothing to do with like some of the things like telomere biology. Their aging has nothing really to do with telomere biology. It's all oxidative stress and mitochondrial dysfunction there. So creating the immortal mouse is something a lot of people are working on. There's even been big prizes offered to anybody that could do it, and the prizes have been awarded. I remember Woody Wright. I actually presented the prize to him. I want to say 20 years ago at a conference where there was a prize given for somebody who's done the most work to make the immortal mouse. And Dr. Woodrow Rice won the award that year. So it's been around for a while. So it's not a new idea.
Mm-hmm. Yes, indeed. Thank you, Bill. Now, in terms of promising trials, now, Liz, I do think the combination gene therapy that you underwent did help to reverse your aging. I'm not a medical professional, but empirically, it seems to me to be the case. So Ampero asks, is anyone else undergoing the same treatments as Liz did back in 2015, or something similar, perhaps with a similar intent? Or are you still the one and only person who underwent that combination gene therapy?
Ooh, that's a really good question. And like Bill said, so when we measure different biomarkers or things that are not considered yet biomarkers of aging, like telomere length and DNA methylation and things like that, we want to track that data because we believe that the accumulation of all of that data together is what's going to make a difference. For instance, maybe just lengthening telomeres doesn't make an immortal mouse. Maybe just partial reprogramming doesn't make an immortal mouse. Maybe combinations of many things, and then therefore looking at many of these potential new biomarkers will make a difference. And so we do that for companies who do medical tourism. We actually step up and will assess and analyze data of any company who's working at a faster pace than considered regular within the United States. So maybe they're working outside of the United States. And we do assess data in which people participate in therapeutics that are not yet offered in the United States around the gene therapy space. And that data is all anonymized to us, but there are several people who have participated in similar therapeutics as I did, and that is the data that we're interested in, and that's the data that we would like to have recognized by a regulatory body in order to get them through a regulatory system to humans. So I am not the only person who has participated in therapies like that.
Didn't you already publish some of that data, or did I just hear you speak about it? Didn't you already publish some of the human data from other patients?
Yeah, so the telomerase reverse transcriptase and the Klotho, that was the dementia study. That was a study that was done specifically with 5 people, and that data is published. And you'll see more data over time as we have larger cohorts. Now, the dementia study that was done by a company called Integrative Health Systems. Now, BioViva, us ourselves, want to see if we can reproduce that study and even do better. And so, we're pretty interested in actually participating in this now, but we have to do it at a different level as BioViva.
Excellent. Well, this is fascinating and great news that other people have undergone these gene therapies now. This progress is not always visible in the news, but it is occurring behind the scenes. So thank you, Liz. Now, our friend Mike DeVerde wonders, what do you, any of our guests today, think of Dr. David Sinclair's epigenetic reprogramming for age reversal? Liz, I believe you mentioned epigenetic reprogramming briefly in your remarks. Would you care to elaborate as to your thoughts on it?
Oh yeah, I mean, of course, I'm really excited about it. Anything that you can do with genes, we're excited because that's something that we can make sure that that type of technology gets to the public at a much less pricey price tag than traditional companies, but we also want to make sure that those type of things don't stay in research. Now, that I consider very new in comparison to the genes that we work with. And so, we wouldn't rush off to humans with that type of technology yet. And so that's why I would rather hear, so there's my opinion. I'm so happy new things are happening in the space. And of course, we want to do some of our own research around that technology. But on the science side, I think you should ask Bill because he would know much better the cellular mechanisms and how close he thinks that is to human use.
Well, we've published a lot on genetic reprogramming and using the Yamanaka factors to do so. I mean, I think since 2008, we've actually been publishing and providing poster sessions. When you use all four Yamanaka factors, you have something occur that doesn't occur with just any three of them, and that's the sudden production of telomerase. Okay, and that might be the only thing, I can't say for certain, but it might be the only thing actually involved in the Yamanaka factors that cause rejuvenation, might cause rejuvenation. Everything else is causing dedifferentiation, and that's the big problem with Yamanaka factors and genetic reprogramming. Is it's more of like turning a butterfly into a caterpillar or a frog into a tadpole than it is actually in making something younger. And I know Basil's just spent a lot of money on trying to incorporate this thing, but I'm really concerned that it's not going to work, and it's actually going to cause cells to dedifferentiate, which can be a problem, instead of just getting rejuvenated. So, but we'll have to see. It's just the best guess on my part. It's just like
I completely agree with Dr. Andrews that I personally, from the research I've seen, I find it too risky to do it in live human beings. So, us. So I find it too risky. And the greatest genetic reprogramming agent we've all known is fasting, especially long fasts. You get the ketosis and the HDAC inhibition. And after 9-day fasts with FGF21 in humans, we don't even know what's going on the epigenetic level. So it's really the final frontier. So if you want to have epigenetic reprogramming that has worked, or at least that's safe to a certain extent, I would just do augmented fasting.
Yes, thank you to everyone who responded, Liz, Bill, and Simon. We have a fascinating question from Jason Geringer, so I hope we can fit it in before we conclude in 6 minutes. Jason, please go ahead.
I was wondering about the really old. I mean, we would have less time to wait. Does this treatment work on the really old, or is there just too much damage? Is that even a viable option, or do you want to start when they're like 50s?
I actually believe Jason's exactly on point. I don't understand why some of the really old aren't signing up for some of the studies like that Liz is conducting. I always say, there's data actually, and I talk a lot about it in my presentations. I talk about sometimes the risk of no treatment is greater than the risk of treatment. And if you are a healthy 89-year-old, you have an 11% chance of dying before you're 90 within 1 year. So being 89 is high risk, and they should be getting the treatments. And I think that's going to allow us to be able to measure if we've extended their lifespan and health span a lot faster. The problem is having enough negative controls, okay? And plus, you might actually have to go ahead and use identical twins to really get meaningful data. And the identical twins have to have been raised 100% alike during their entire life. And so those studies become very convoluted, I guess is the right word, because no matter what results you get, somebody's going to find some reason to say, well, it wasn't properly controlled. But yeah, I'm dying to see some of the, like I've got 95-year-old people calling me all the time that want to get treated, but they just never get around to actually saying, okay, here's the money to do it. And I just don't understand it. If I was 95 years old, I'd be the first one in line to get treated by Liz Parrish. But I think there's a chance, but I can't guarantee it. I just think there's a chance that one of the future modifications of the procedure is going to actually, that Liz is doing, is going to actually turn a 95-year-old into a 25-year-old. I think that's a chance of that. I'm not saying I expect it, but I wouldn't be surprised if that happens. I think there's opportunities.
Wouldn't that be
Maybe
awesome
I require
to
all
see?
3 or 4 of her genes, but otherwise, wait, we're running out of time. Go ahead, sorry.
Yes.
That would be
So I
awesome.
would love to get an alpha clodo gene therapy from Liz. So I'll be the first one in line for that one if it's going to humans. So let me know about that.
It is right now.
Yeah, thanks. Yeah, I actually tried alpha clotho in 2020, and I look forward to doing more. But I'm not saying that you should do it. I'm saying that you should have the right to do it if you need to, and certainly terminally ill and very old people should have the ability to participate in these therapies if they are so inclined, or if they have someone who has power of attorney that is so inclined to help them get access to those technologies. Would it work in a very old person? They have a lot of damage. It's probably, I mean, it's like a guess. It's probably a lower likelihood that you would have a positive impact, maybe, but it has to be done. And I don't think that there is an honest to God, true scientist out there that makes speculations. So, you know, the people that I work with are people who are asking the question, but they're not answering it with their ego. We have to answer it with human data.
Yes, thank you very much. And I would like to acknowledge several of our very generous viewers, including Celeste Castaldo, who says, thank you for the great discussion today. She also points out an achievement of gene therapy in California, stem cell therapy and genetic medicine, where bubble baby disease has been cured. So that is quite fascinating. We also have a comment from our friend Alan Crowley, who writes, fascinating discussion today, even better than usual science and policy. And he says, if you need a test subject, I'm available. Keep up the good work. And we've had several others make comments along similar lines. Our friends Mike DeVerde, Edward Hudgens, and Simon just now. So it looks like there is ample interest in these emerging treatments, and I'm sure among others in our audience as well. We have 1 minute. David Shoemaker, would you like to make some brief closing remarks? And I'll give the same option to Art Ramon as well.
Yeah, I'll be characteristically very brief and just say this has been a fascinating discussion today, and I've been most interested to take notes and look for different areas to do different research. So thanks for our guests that came today.
All right, thank you, David, as well. And Art Ramon, would you like to say anything?
Yes. Have there been any studies that compare the difference between the mitochondria in a female ova, like right before menopause, and the rest of her mitochondria to see what the differences are in that mitochondria, the damage, and the ova, which starts off the newborn having, fresh mitochondria? So any studies like that?
I think this is a good question for Dr. Niezen-Koning. Are you aware of anything along these lines? And just a moment. I think it would be good. Yes.
I was muted. I'm not aware of those studies, but we have performed ourselves some pyrosequencing studies in urinary mitochondrial DNA with pyrosequencing, as I told you, and we saw an increase in methylation during time. So we had young children and we had old females. It was all females, and we saw an increase in methylation. Is this question answered?
Yes. It looks
Okay,
like
thank you.
Art Ramon has been satisfied with the answer. So thank you very much.
Let me add,
And
Art, there's a lot of stuff in PubMed. A lot of studies have been done like that. Just go to PubMed and search. You'll find tons of studies about mitochondria and genomic DNA.
all right, wonderful. Thank you very much to all of our presenters today. Thank you to our panelists for the excellent questions and interactive conversation. Thank you also to Dan Elton, our Director of Scholarship, for his generous super chat. And as he says, keep up the good work, everyone, and live long and prosper.